since Jeff isn't here, I'm just going to talk about sort of one more
Oh,
aspect.
Florian, flip the on-air sign just as a precaution. Thank you.
3, 2, 1, let's go. Huh? Full Numenta? Oh my God, yeah. Pretty full Numenta, yeah, exactly. Why don't I have Numenta?
Now, we need Numenta suspenders.
Numenta suspenders.
Like, I don't think we can all come dressed as Florian.
Wouldn't that be fun? All right. So I guess, the proper title for today's talk is going to be synaptic depression. And I'm going to specifically talk about the so-called Tsodyks-Markram model, which is the most common model for accounting for synaptic depression in people who are simulating spiking neural networks.
What is the name again?
So Tsodyks-Markram, right? So Barak Tsodyks, who's actually he just joined some American faculty. I don't know where he went. He was Israeli Weizmann Institute, where Henry Markram was also earlier, before he started that whole blue brain, human brain project stuff. So maybe first we should talk a little about, I mentioned last time I was talking about facilitation. Now I want to talk about the flip side of that, which is synaptic depression. So I hopefully conveyed last time that there's an important effect that successive trains of spikes can increase the release probability of synaptic vesicles. And it has a lot to do with calcium dynamics, and there's a lot of things at play. But the main point of it is that a synapse can go stronger just because it has been used before. So just the fact that there was a spike 10 milliseconds ago will make the next spike a lot more powerful in terms of the effect that it has on the postsynaptic neuron. The reason for that is that you are more likely to release vesicles. That effect is called facilitation or augmentation, depending on the time scale, or post-tetanic potentiation, if you're talking about 100 spikes put on there and then some transient effect.
And importantly, that's a short-term effect.
And importantly, these effects decay away. So if you just, sprinkle some random activity or you just wait for long enough, the neuron will return to where it was. Today, I'm going to talk about a different short-term effect, which is necessary to introduce for a complete model of short-term plasticity, which includes both, namely synaptic depression. The idea behind synaptic depression is that if you look at different neurons and you do this thing where you put a spike train on it, sometimes you see facilitation, but sometimes you see the opposite of it, where when you put on a spike train on a neuron and you record the postsynaptic membrane voltage, so you're measuring the effect on the postsynaptic side, then on the first spike, you see this very nice, big deflection. That is your excitatory postsynaptic potential, which decays away with a time constant of maybe 5 milliseconds. And then there's a second spike comes in, and then for some reason, right, that second one tends to be smaller and even smaller. Since this occurs typically at very high rates, oftentimes this looks a little bit different. What it actually looks like is more like this. So here you would have an attempt to transmit, the pre-synapse spiked here, 1, 2, 3, 4, 5 spikes. So that was a brief burst that got transmitted from the presynaptic neuron to the postsynaptic neuron. Again, keep in mind, right? Synapse, vesicles being released, right? Opening up into the synaptic cleft, spilling their neurotransmitter, and then you have the postsynaptic side with the receptor, right? Where there's docking sites for the neuroreceptor, which then, exchange ions to change the membrane potential here. This is the membrane potential we are measuring here, right? So there's there's an electrode here, right? Patch clamp ideally. So it actually looks a little bit like this, right? Kind of patched in there. And then this is what we're recording. And so what you are seeing here is that the first excitatory potential is very large. So this might be, you know, maybe that's a millivolt. That's a rather strong synapse. And then the second one is actually a lot lower. And it is already lower because the membrane is more depolarized. So it's harder to push ions into a neuron, right? Because its voltage has already changed towards the firing threshold. But it's also smaller because the effectivity, like the effect of the synapse actually goes smaller and smaller. So one way that people make sure that this effect of smaller and smaller excitatory postsynaptic potential is not because of the membrane voltage, is they clamp the voltage down on this side. So they actually force the neuron to be at this one particular voltage. And then instead, they measure sort of the deflections on that, right? So that you essentially force this back to the ground every time. But then you still see the same effect. So it's still true that the synapse tends to weaken every time it is used. This effect is called synaptic depression. And so whereas facilitation was an increase in release probability, depression is commonly interpreted as a depletion of the pool of readily releasable vesicles. So there's some vesicles that can be released by a spike, and if that doesn't replenish very fast, and if there's no facilitation, so no mechanism to make it replenish even faster and release, you know, more every time, then instead, what you will, instead of a strengthening that you saw last time, you're going to get a weakening of the synapse.
The question, so there's a big difference between these two curves.
Mm-hmm.
If I understand it correctly, the impact of each additional presynaptic spike is smaller, but overall, the impact, if you look at the peak of that top curve, is still higher than a single spike would be.
Yes, you still get a summing, right?
Like maybe a 2 millivolt or something.
Right, so 5 spikes deliver you an extra 0.5 millivolt there.
You don't get 5 millivolt, but you get,
right,
it is more than 1 millivolt.
Right, exactly. So you
Still
get
a bigger effect.
Yes, so the synaptic depression does not fully cancel out the effect of a burst, but it means it's not linearly summing. Again, right, we had superlinear summing before with facilitation, where every spike becomes more powerful. Here it's the opposite, every spike becomes weaker. And so all I want to introduce you today is a mathematical model built by Tsodyks-Markram to explain what is happening here, right? So they introduced this notion that there is a synaptic resource. This resource is the number of releasable vesicles. And I already talked about the fact that there might be some fraction of vesicles that might get released, right? This is what they call this USE, right? So the synaptic efficacy. So it's a utilization of the synapse. So what percentage of releasable synapses get released when a spike occurs? Say that's some number, 20% in like a typical cortical model that I use. So the argument goes, when there is a spike, 20% of the vesicles get released, which means that afterwards this synapse is a bit weaker now. It only has 80% of the resources. So if the next spike releases another 20%, that is, less, even if that number stays constant. And so this is what you see in this equation here. So X is some kind of a resource, right? You could think of this as the reserve pool. Let's maybe actually write this out here. It might actually help, because obviously X, Y, Z is rather abstract.
X, Y, Z seems to denote coordinates of some sort.
Yeah, exactly, right? So this differential equation, the idea is that you have some pool of inactive from which you can recover resources, because of course the idea is that you get to replenish either from a background pool or even from recovering transmitter to put them into new vesicles. You get to replenish the supply of resources. So there's a recovery time constant. So how long does it take until the synapse is as strong as it was before on the first spike? For typical cortical model neurons, for layer 2-3 that we're using, that's on the order of like 500 milliseconds. So after a second or so, if you separate your spikes out and you only have one spike a second, 1 Hertz activity, then this will very nicely be 1 EPSP every time, and it's going to look the same every time. if you go faster, you're going to run into this problem that there's some percentage of the available resources at the time of the spike that will get used up. This is a delta function. So that function is 0 at all times, right? Except at the spike time, TSP, where it is 1. So when there is a spike, some fraction, so let's say 20%, of the available synaptic resources of the reserve pool will get used up. That's why it's subtracted. That pool of resources will go into a second pool. So you see that transfer there into this Y pool. This is the pool of active neurotransmitter. So you can think of that as the total amount of released transmitter here, right? That is out in the synaptic cleft. So maybe it might be useful actually to put this in here. So to say this is sort of your reserve. This is the let's call it active part. And then there is also the inactive part. So this pool gets depleted by some percentage when there is a spike, and it recovers back with some time constant from the unused transmitter. The active pool is actually acting on the synapse, and what it does, it activates receptors, and when you activate a receptor like that, this is pretty fast, right? So there's no time constant with this. It's like an instantaneous jump at some delta t again. So it's the same delta function, and it jumps up by the exact amount that just got taken away from there. So, but then there's some inactivation because this transmitter is not effective for all the time because it eventually undocks, and these synapses close, the receptors close. The time constant on these decays is on the order of 5 milliseconds. So let me put that in here too, these numbers. So this is on the order of 500 milliseconds, and this inactivation is on the order of 5 milliseconds. So the recovery is a rather slow dynamic. The using for actually affecting the post-synapse is actually rather fast, right? But then this is gonna, decay with this fast time constant, so the receptor becomes kind of used up, right? You can think of it that way. And then the idea is that this used up transmitter then goes into this pool of, well, inactive transmitter. And then, so that's why, right, this is a plus here. And then this one recovers with that exact time constant we already talked about here, right, back to here. So you hopefully see that this equation system is a closed loop. So total amount of transmitter that is going around stays the same. And what you're trying to account for here is the temporal dynamics in a network where you have neurons that you know their spike times off, you know when they spike, and you can try to fit these parameters, right? You can try to fit the depression time constant, the synaptic use, so what's the percentage of transmitter that gets used up with every spike, and you can try to fit this postsynaptic time constant, right, which is also called like the synaptic trace. This is actually different for different transmitters. So the really fast one of 5 milliseconds, those are the AMPA receptors, which are the most common and powerful glutamate receptors that mostly drive the membrane potential. But there are other ones. There are these NMDA receptors, which you might have heard, which have a lot longer time constant. That's more like on the order of 100 milliseconds. There tend to be fewer of those. So if you were to plot the contribution of the NMDA receptor, it would look a little bit different, right? It would like be less of a peak height, and then it would be a very slow decay, which means they actually sum up a lot better over bursts than the AMPA receptors. So the AMPA receptors are for the fast transmission, whereas the NMDA receptors give a sort of longer lasting trace. So you can actually have different receptors in this model that use a different time constant to account for not just the dynamics on the under one type of receptor, but under a pool of different types of receptors.
They would all be using the same set of neurotransmitters.
There would, yes,
Yeah,
because
so
NMDA and
this-
alpha both use glutamate.
Yeah, so this Y and stuff would be shared
Yes, exactly.
between these different
Right.
receptors.
But you would get different contributions, right? So to the postsynaptic membrane voltage. And so, so I just simply wanted to show this model. Obviously, what we have not talked about yet is the facilitation part. So, because the idea behind facilitation is that this number, the 20%, can actually change. In facilitating synapses, this number goes up, right? So it's, the first spike maybe use 20% of the resources, and then the second and third spike both use like 30%, which means that, you will also, either need to speed up the recruitment of reserves somehow, right? Or your synapse is gonna get like depressed very quickly. Typically, most neurons in cortex are classified into either facilitating or depressing. And what I will talk next time about in the context of this working memory model that I'm going to present is how useful it is to have networks that have both of these, like depression and facilitation, because you can then use them as working memory models in a very interesting way.
Two questions.
I can ask it.
The subscript rec, is that for recovery or was it simple?
Yeah, that's recovery. Yeah.
And I assume second delta one would be T zip SP.
That is correct. Yes. You see, that happens when you don't take time that markers to neatly prepare the talk. Exactly.
I have a layman question. So the neurotransmitters go from reserve, tractive, to inactive. That's the flow. They never go into the other. I don't know even the words. They never go into the other spine or gluton.
Oh, you mean whether transmitter ever goes in here?
Yeah.
I'm not aware of an uptake mechanism on the postsynaptic side. I've never heard of such a thing. What does exist is, of course, glia cells. So there are cells, that are not neurons, right? And they do some recycling of receptors. They're like maintaining a balance. So you can show that when you have too much transmitter here, actually some of it is not taken up by the presynaptic neurons to refuel vesicles, but actually there's some kind of moderation of the level of sort of the synaptic, available transmitter also by these glia cells. So it gets more complicated, but as far as I know, not on the postsynaptic side.
The one on the top is spine, the one on the bottom is a gluton. What is it?
Yeah, I think the one at the bottom is the spine.
Oh, okay.
And
The one who releases?
Right. I mean, it's an axonal release terminal, right? Where you.
sometimes glutons.
Yeah.
Okay, so gluton on top and spine.
Right.
I think maybe the one thing in your question is these neurotransmitters, their function is to kind of bind to these receptors, and then it'll open up and other ions will go into the cell. So
Yeah,
the neurotransmitters are not going into the cell, but they're opening up the channel to let these other ions into the cell.
All right.
Yeah.
Okay.
So
Yes. I was wondering how that works.
Exactly, that's the-
okay.
Right.
So that's what increases the charge or decreases the charge.
Okay. That makes sense.
That's where those deflection comes from sodium and potassium changing sides.
Okay.
Right? And that changes the membrane voltage. So like for one molecule of transmitter, you get hundreds, thousands, I don't know, maybe tens of thousands, I'm not exactly sure of the numbers, of ions flowing in through that now activated receptor, which is why there's quite a change, right, of like a full millivolt or something in a strong synapse. Most synapses are weaker than that.
And that doesn't depend on the concentration of ions in the.
Yes, of course it does, which is why that is carefully mediated in the cerebrospinal fluid. The balance of the different ions is carefully maintained by all kinds of mechanisms.
Or you have the neurotransmitter, but you don't have the ions that won't work.
Right, exactly. Yes.
So doesn't that influence like the equation?
Yeah. You could do that, right? You could have a solution where you exchange some ions out. So, for example, in the facilitation experiments that I talked about last time, what you can do is you can exchange the calcium for magnesium, which is like they're chemically similar enough that you can replace them. But then all the mechanisms for facilitation that rely on calcium flowing in, they won't work because there's very little calcium to be able to flow in. Actually, the calcium concentration might be higher inside than outside, so maybe you get an outflow of calcium even though there's very little calcium in the cell. And then you can show that there will not be facilitation, which was sort of one incidental proof that facilitation is depends on the calcium. So sometimes you can exchange ions. It's very tricky to replace sodium and potassium. You can't really remove it fully from the fluid because the osmotic pressure would be so high, like your neurons would burst.
That actually was one of the questions I had. So you said there were two mechanisms for the depression. The second one, which you described, was the depletion of vesicles. But the first one seemed to imply that when you're trying to get more charge in, obviously existing charge, and there you're pushing against
Right.
So there, in my mind, there's two properties there. One is pure osmotic, and the other one is actual charge field. I'm wondering to what degree those should be treated separately.
Right. That is a really good question, and that is that the problem with that is that it really depends on like the size of the pulse neuron, and how quickly ions move around. Right, these ions move within milliseconds to all kinds of places, but the total volume matters, right? So you can think of that as like the capacitance of a neuron. If a neuron is very big, then it can receive lots and lots of inputs before it changes the potential. Meaning you will get a lot of full-size DPSPs if there is no depression, because the voltage won't move much, right? Whereas in a very small neuron, like if the postsynaptic side is very small, right, there's very little capacity to take up new ions, right? Then a couple of ion pumps can very quickly change the membrane potential, and then you're already very close to the firing threshold, and then it's really, really hard to make more ions flow.
That's straight.
Right. So it's really what the balance between all those is really depends on the physiological properties of these cells. I am obviously most familiar to the numbers in standard, layer 2, 3, sort of small and larger pyramidal cells in the supragranular layers, because those are the ones that I've modeled extensively. Yeah, again, different neurons are different when it comes to this.
I understand it, guys. It's also a matter of active debate in the community. In vitro, you can set these concentrations to whatever you want,
right? And
the potentials, and people set them all over the place,
right?
But in vivo, what is the actual potential difference? And it's actually not clear.
So a lot of people are obviously a lot of work has gone into manufacturing the perfect artificial cerebrospinal fluid that has the exact right composition, and there seems to be, like, as far as I understand, like a pretty good consensus and on what situations and what the temperature should be, obviously, because if it's all 5 degrees lower, then the neurons might not be active at all. And the problem is always with in vitro, of course, you're cutting all the inputs, so like the whole way that the network behaves is very different.
Because in vivo, there's lots of other inputs coming into the cell, and so the actual potential difference is actually hard to determine.
Yes. But you can still do experiments like with patch clamping in vivo, right? So you can, in a head fixed, mouse or something, right? You can apply, patch clamps, even to both sides and have like full control over what happens. So it's not impossible to investigate this, but it's hard to do. It's hard to do it in a way that, gets all the possible ways that people might criticize it right. But the important thing is that, even in vivo, these effects are real, and both facilitating synapses as well as depressing synapses exist. And obviously, depending on the neuron, depending on the receptors and the types and where in the brain and whatnot, all these numbers are going to be a little different. The value of the Tsodyks-Markram model and what people have done with it is that the tuning knobs that it has tend to be the most useful ones. So you get away with just adjusting 4 or 5 parameters, and then you have a pretty good model. Whereas if you do a recording on a simulated neuron and a recording on a real neuron, the spike trains look rather similar. which is the whole point of these models, right? You want to capture the evolution of the voltage, right? Because the idea is obviously that underlies information transmission. That is all I had to say for today. So,
can I ask something? Yeah.
Mhm.
I want to see if I'm oversimplifying facilitation and depression by intentionally simplifying it to say, like, from an information processing standpoint, here's one narrative you could put on it. And I'm going to sort of let's see. I'm going to do a 2x2 matrix of facilitating versus depressing, and we'll say one spike, one presynaptic spike versus like multiple,
right?
Like
a burst.
Yeah, multiple spikes. And we could say, just like, I'm going to oversimplify it and just say a facilitating synapse has like a weak, we'll say strong, and I'm just going to say strong.
Is that true?
So one narrative you could put on this, I'm just going to ignore the fact that this gets slightly stronger. I could put like a strong asterisk or something like that. But like one way you could choose to think about all this is just a different threshold. A facilitating synapse is a synapse that has a higher threshold. It requires multiple spikes to get its true response. A depressing synapse is just something that a single spike will basically get it to its threshold. It's like a noise tolerant synapse versus a very sensitive one.
Right.
It's still stronger in the multiple case.
Hence the asterisk, slightly stronger.
At least 50%.
The relative contribution.
But I mean, you were just saying the in vitro and vivo thing. Is this just something that happens when we're patch clamping, or?
No, this effect is really, I mean, it's well understood and it's
like
real,
the depressing part happens in vivo.
Right, and it simply has to do with the fact that the amount of releasable vesicles like goes down and is not replenished fast enough. And unlike facilitating synapses, there's no mechanism to speed that up.
So am I oversimplifying by just saying maybe it's just thresholding, a threshold difference?
Why do you say that facilitating is weak for one spike? Is it a lot less than a millivolt?
No, so I guess it's not really fair to say, because there are, of course, synapses that are neither facilitating nor depressing.
Okay, but that's a separate.
So there's the third category called neither, right? I'm not quite sure how useful this is, because depending on the neuron and the synapse, all of these can take all kinds of forms. Yeah, it might be true. I don't know that facilitating synapses are on average weaker on the first spike, but I don't know that to be true.
Okay.
Because I know, for example, non-facilitating ones, also non-depressing ones, let's say the big basket cell, paravalvumin positive cells, right, that do this big, powerful inhibition in the local microcircuit. They tend to put out the same, IPSP, so it's inhibitory, right, on every burst at a very high rate, like fire 70 hertz, no problem, or even higher than that. So they must have like either a really big pool or that gets carefully released, or a pool that's capable of like very quickly regenerating. So it has an internal mechanism, machinery, without actually increasing or decreasing the speed at which that happens. So they're neither facilitating nor depressing, and they're very, very strong. So
excitatory synapses, are there categories like that?
It's a great question. I would be hesitant to say, straight up yes to this, but it is an interesting idea. Right?
So question I had was the layer 4 pyramidal cells, when they get direct thalamic input, the relay cells. My understanding is those synapses are depressive synapses. And so I was actually trying to understand the stronger issue for that.
Right.
Because it relates to some of the stuff we've been thinking about the thalamus and the tonic spike versus the burst of spikes coming in.
Yes.
But the burst, if I understand it, should still give a stronger result. It's just that it's capped.
It's
linearly increasing.
Yes, sublinear, right?
Yeah.
It's sublinear summation as opposed to facilitation, which is supralinear summation. Right?
And then do you know how much stronger it gets? Is it 50%? Is it twice as
I can tell you from,
what is the peak of that?
obviously it depends on the balance there. There is some saturation, right? So even an infinitely long train of presynaptic spikes will inevitably arrive at some point where there's a balance between the input and the depression. So at which the voltage that you get really only depends on what is the speed of recovery here, right? And the time it takes to get to that point depends on your usage number, right? If you're releasing very, very little transmitter at every spike, then it's going to take a long, long time until you reach that. So you can sum a lot of pulses, right? But they're going to be small. If you have big pulses, then you're going to deplete, maybe within 3 spikes that pool. So you're almost at the, limit already after 3 spikes. That tends to be different in different neurons, and depending on the circuit, the connection you're looking at, you need to choose the right parameters. Since you concretely asked, I've done obviously a lot of layer 2-3 work, and the interesting thing that we've been looking a lot are so-called theta nested gamma bursts. So tend to be like clusters of like 5 spikes at 60 hertz or something. It's like very commonly seen in prefrontal cortex. So we used it as a PFC model. And those tend to sum up, I would have to look into the Journal of Neuroscience paper that I wrote, but I would venture to guess that like the 5 spikes add up to, maybe double the first deflection. So instead of 1 millivolt, I get like a total of 1.8 or 2 millivolt deflection.
So it'd be twice. So with multiple spikes, you get about twice the effect.
Right. And if I double that again, I would not get double. I would get, another 0.3 millivolts or something.
Because these thalamics, thalamocortical synapses are pretty large. Right. Also, so
one thing I should mention, because of course people wonder a lot about how to understand this in information theoretical terms, like what is the information theoretical function of this? The way that people build this table instead is they talk about the usefulness of these synapses for transmitting either rates or spike times. So the idea is that facilitating synapse, right, will sort of become active when there is a whole rate of pulses that needs to be transmitted, and when, let's say, the length of a pulse matters. Then you really want a synapse that is capable of staying up and long, like recognizing sort of the beginning and then becoming powerful so it stays on for the whole time. Whereas if you want a synapse that can measure, that can transmit in information theoretical terms an exact onset of something, like in precise timing, like you see a lot in auditory processing, where the exact spike time matters a lot as opposed to the rate, which is not all that important, then you really want a depressing synapse. And true to that, you find a lot more depressing synapses in circuits that are, need to process exact spike times, as opposed to circuits that transmit, long trains where like there's some firing rate that transmits information. In which case you don't want a depressing synapse, because after a short amount of time, it's going to be useful for transmitting the rate. So then you rather want either a non-depressing one or or even better, a facilitating one, right? That has active mechanisms that help you transmit a firing rate on the, to the postsynaptic side. So instead, right, the categories are instead rate coding, and time coding. And then the category is essentially that facilitating synapses are good for this, and depressing synapses are a lot better for this. And then, it gets sort of like tricky, what do you do in these cases where a depressing synapse now is subject to a long rate of firing, right? Because obviously it's going to kind of shut off after some time, or its transmission will flatline, right, at some jump that just depends on the recovery. So that is typically how people then look at it in information theoretical terms.
There's those excitatory synapses that we think about with these active dendritic segments. Are they facilitating?
That is a great question. I don't know. I would, just because they are inside like supragranular layers, I would have a hunch that they're probably more depressing, facilitating.
So they'd be in every neuron. It's not specific to layers. It's just of the distal, the 10
Right.
dendritic segments.
Right, but this tuning of these parameters tends to be a little bit different in pyramidals in different areas. That's what I'm saying.
Okay, I guess I was just wondering whether these active dendritic segments where you have NMDA spikes.
Yes.
Do you know if they happen to be depressive synapses or?
I don't know for sure, but if I had to
You
guess,
would guess
I'm pretty
depressing.
confident they're depressing. So they would detect the coincident activation of something in some short time window, and that's what they would be good at. So it would be a lot more, detecting some specific time at which certain units are coactive, rather than transmitting a constant stream of inputs forward to something, which would make sense also for the Numenta model. Obviously, I could take a look at the difference between basal and distal ones, whether there's people who've specifically looked at this balance.
But then that kind of begs the question: if the proximal ones from thalamus are depressing, and if you say the active dendritic segment synapses are depressing,
right?
Where are the facilitating ones?
Layer 5 is facilitating ones. There's across cortical layers, like 2, 3 to 5, I think, are facilitating. But again, I can give you a list. I've taken a look at those. People have been trying to catalog those. They matter obviously for biophysical models where you have spiking neurons, because if you don't get this right, the transmission of information is going to look very different, right? Because it's like between facilitating and depressing, when you're transmitting a gamma burst, the difference can be like an order of magnitude in terms of what is perceivable on the other side. Right?
How artificial is that boundary? Like a hard binary? You have cell A, cell B, but is there a continuum and someone just put a line in the middle and said, let's call this facilitating, let's call this depressing?
There are established experimental protocols that sort of define this. The idea typically is to hold the postsynaptic neuron constant, and you measure the current that is needed to keep that constant, right? So you have like an electrode that can insert some current. Let's call it I post. That is the current that is needed to keep this membrane voltage at a constant level. So that way you avoid the problem of osmotic pressure, changing your membrane voltage, and thus
you're driving charge.
Right. So whenever some EPSP happens here, instead you're not going to measure this thing, because that's going to be canceled out by some controller. Instead, what you're going to measure is the current that is needed to rebalance the synapse, right? To, I don't know, minus 70 millivolts or something. So you keep that at that value, and then you measure that current. And typically people just use pulse pairs. So you just transmit 2 spikes at some defined frequency, like some interspike interval. And then you just show, did the current that got transmitted from the first spike increase in the second spike or decrease? If it increases, it's a facilitating synapse. If it decreases, it's a depressing synapse.
It's more like a hard one.
Right, which is pretty, that's how you define it. Obviously, there might be neurons in which it is more complicated, where like you get some facilitation early on, but later on it actually depresses. So like, where like the effective EPSP first, increases 40% and then it starts dwindling down again. So, but generally, at least my impression is that neuroscientists are very confident putting all of the neurons kind of into 3 buckets, sort of facilitating, depressing, and non-adapting.
Does a neuron potentially serve multiple roles over like different periods? Like could be facilitating like over like some stretch of time and suddenly change into.
I mean, whether there's some neuromodulatory thing that could change a synapse to become facilitating, or to become depressing, or that is a good question. I'm not aware, but maybe there is some cool mechanism that does that. That would be new to me though.
One thing I'm noticing there is that you've got 3 state variables and 3 parameters. Again. So I'm assuming that in those time constants, all the details of metabolism and you're doing active work where you're doing this stuff. So obviously energy is being expended, is being generated. All that's kind of folded into that time constant.
Yes.
Question in my mind is that really a constant? Because it would seem like if you run out of ATP and you can't drive things around,
Right.
then you would have another way of depleting, the ability of the synapse to do work.
Right. That is a good question. It's true, once you dig down on the biomolecular mechanisms behind that, it's not one process with one time constant, but it's actually many processes, and some are faster and some are slower. And the time constant is just a good approximation for the kind of timescales that we are looking at. Obviously, you can have absurd scenarios like an epileptic seizure, where suddenly you have 100 hertz activity for seconds long, right? Which is gonna depress every synapse, at least in the supragranular layers, right? So you're literally exhausting that pool to its maximum. And obviously there's huge energy demands then to recover that, to make that possible. And obviously that's not compatible with active information processing, right? I always like to joke to people who tell me this weird story about, oh, can we increase the percentage of brain activity, to make us smarter or something? Yeah, I've seen people with 100% brain activity. It's not a pretty sight. I learned how to put people into a stable side position and to help others 3 times in my life. So yeah, it depends. Again, these fits are typically made for timescales and firing rates that are within the realm that is sort of plausible. And then, of course, you can always quibble whether it would be better to have 2 time constants, one for a faster process, one for slower, but it gets very complicated, right? The idea of these models is to find a neat expression with very few variables that explains a lot of different spike trains, a lot of different synapses, a lot of different circuits that can be mapped onto this if they have depressing synapses.
The hope would be, I guess, is that the design of the synapse is that you don't run out of secondary, tertiary resources in order for it to do what it's supposed to be doing.
Right.
Except in pathological conditions.
Right.
Do you have an idea on the percentage number, the percentage of cells that are facilitating and depressing? It's supposed to be
Is it a cell or a synapse?
Oh, sorry, synapse.
Yeah, that's a good point,
actually.
right? That is a good point. The facilitating or depressing property is a presynaptic property, right? Because it depends on whether the calcium in here will make the release probability higher when they're facilitating, and it depends on how quickly this reserve gets replenished on the presynaptic side, the mechanisms for recruiting the reserve pools into actively releasable sites, and how much gets released every spike. So all of these are presynaptic properties.
But the same axon could have a facilitating bouton and a further away depressing bouton.
Theoretically, practically, I'm not aware of that.
Okay, so a cell is going to be either
facilitating,
really.
right? So for example, basket cells, right, make powerful inhibitory connections. All of those tend to be very stable, high rate, fast spiking, tend to transmit fast spiking inhibition, which neither depresses nor facilitates a lot, meaning you get a very powerful and very constant fast feedback inhibition. And that doesn't change. And that's the same at all the synapses that basket cell makes. So generally, that's why this is a property of the whole cell that applies to all of its.
What about a pyramidal cell? Mm-hmm. All the connections it makes is going to be either one or the other.
So that goes to the fascinating question: are there modulators to this? Right? I am not aware of any, but of course, when you're talking about biology, if something theoretically can be conceived of, there's probably some case in which that is the case. All the models that I have seen typically assume that all of the synapses that a cell make have the same presynaptic properties with regards to facilitation, depression, non-adapting.
So what are usually pyramidal cells? Are they usually facilitating or their synapse?
So most of pyramidal cells tend to have both exist, and it depends on where you look. I'd be hesitant to give a general answer, and I don't know the exact ratio how it breaks down.
In the cortex, both exist, both of these.
Yeah. All right. Good. Then that was that. So next time I'm going to talk how to drill this open, because this number, this 20%, might not stay constant. It might also be actually temporally varying. So one more differential equation. Turns out when you do that, you can then collapse this, so it boils down to 3 equations again. So don't worry about it. It doesn't get too messy in terms of the math. But then you can have a general model that, depending on your parameterization, can be facilitating, depressing, a little bit of both. And you can model a giant zoo of cells with that, which is awesome. And that is then the full Tsodyks-Markram model of the 98 paper that they published. And then show how to use that for working memory. Good.
Cool. Thank you so much. That's
Thank
really
you.
great. Thank you.
Thanks, Florian. I'm gonna take the live stream down now. All right. Thanks everybody for watching. We'll be back on Wednesday. I'm pretty sure we'll be back on Wednesday. Jeff has been out of the office. He's got a personal thing that is keeping him away. But hopefully he'll be back soon. I thought he would be back this week. So Wednesday I'll be talking about the affordance competition hypothesis. I would really like Jeff to be there, so if he doesn't make it, I'll probably put it off again. But that should be an interesting conversation. That's work from Paul Chisek from, I think, the University of Montreal. I've been reading a couple of his papers. They've been popular conversation topics on our forum as well. So check out our forum, HTM forum, and there's discussions of affordance competition there. And see you on Wednesday. Thank you again for watching. Don't forget to subscribe to the channel, like this video, etc., etc. Take care and have a wonderful day. Now I have to find the button that turns this off. There it is. Bye, guys.